Hello, and thank you for joining us for this Agilent Technologies Cell Analysis webinar series. My name is Ian Hayes. I am the Pharma Market Manager within the Cell Analysis Division at Agilent, and I will be your host for today's webinar. Before I introduce our speaker for today, allow me to share a bit about Agilent Technologies and the products we offer for cell analysis. Agilent is a trusted leader in life sciences, clinical research, diagnostics, and applied chemical markets, providing laboratories with instruments, services, consumables, applications, and expertise to enable our customers to gain the insights they seek. Agilent's rapidly growing cell analysis portfolio enables deeper, more realistic insight across a variety of cell analysis applications, where investigators and drug developers seek to understand complex cellular environments and interactions. This is critically important in the large and growing immuno-oncology and immunotherapy indications. I'm delighted to introduce today's speaker, Dr. Sandra Milasta, Senior Scientist for Elevatebio, Waltham, Massachusetts, to present on the fascinating and topical subject of metabolic regulation of adoptive immune cell function. Sandra received her PhD from the University of Glasgow in the UK and charts an impressive career from academia into industry, with extensive research experience in T and B cell biology and a core focus on immune metabolism and mitochondrial function. Sandra spent 12 years at St. Jude Children's Research Hospital in Doug Green's lab before joining Bluebird Bio and then most recently Elevatebio, where she leads the implementation of processes and experimental platforms to develop T-cell products and supports the company's immunotherapy programs. Before I hand over the mic to Sandra, I would like to remind you that this webinar is interactive, and there will be a question and answer period immediately following the talk. You will notice on your screen that you can, and in fact should, type in your questions during the presentation, as this helps to ensure that we get as many of your questions as possible. With that, thank you, Sandra, for joining us today, and please take it away.
Thank you, Ian, for this really kind introduction, and I'm really excited that Agilent gives me the opportunity to talk a bit about my work on metabolic regulation of adaptive immune cell function and the wider implications of those research efforts for cellular therapy development. I just have to start with a quick disclaimer that even though I'm not working at Elevatebio, I'm obviously expressing views that are my own and are not necessarily shared by Elevatebio. It is widely accepted now that T cells undergo metabolic reprogramming in response to activation. Naive T cells are considered to be metabolically quiescent with only basal nutrient uptake to support their maintenance. And they break down fatty acids, glucose, or amino acids to fuel oxidative phosphorylation. In response to activation, T cells undergo metabolic reprogramming, and now they switch from oxidative phosphorylation to aerobic glycolysis. And this metabolic reprogramming is essential for them to supply enough biosynthetic intermediates for the synthesis of lipids, nucleic acids, and proteins to support this exponential growth and proliferation, as well as to maintain the redox balance of the cell. Memory T cells are now considered to be metabolically primed, and similar to naive T cells, they again rely on oxidative phosphorylation to gain energy, but this is now mostly fueled by fatty acid oxidation. They also have an increased spare respiratory capacity as well as mitochondrial content, and it's the thought that those features enable the T cells to survive long term as well as to quickly respond to secondary challenges. So to add another layer of complexity, individual T cell subtypes also have a unique metabolic phenotype to support their specialized function and maintenance. So for example, Th1, 2, and 17 T cells drive energy via glycolysis, whereas regulatory T cells rely on oxidative phosphorylation fueled by fatty acid oxidation. Dysregulated immune responses are the hallmark of many immune-mediated diseases, including autoimmunity and allergy, where obviously excessive immune responses have very detrimental effects. On the other hand, the generation of a robust and stable T-cell response is the goal for any vaccination strategy as well as in immunotherapy. So therefore, the idea that by targeting metabolism, you can drive a desired T-cell phenotype and thereby improve current treatment strategies for therapeutic or immune-mediated diseases is actually very appealing. In a healthy person, the T cells exist in an environment that is provides sufficient oxygen as well as nutrients that are essential for proper T cell functions, including glucose, glutamine, and arginine. Therefore, enabling the generation of a robust effector response and antivirus immunity upon encounter of a virus. However, in the tumor, the T cell encounters a very different environment, and especially in the solid tumor, low oxygen concentrations lead to hypoxia, and the T cell now also has to compete with the tumor cell itself for the available resources. And more often than not, unfortunately, the T cells lose out to the tumor cells, and this now drives suboptimal effector responses and often leading to T cell exhaustion. So I therefore became interested really early on to understand if we can improve the anti-tumor response by manipulating the metabolic pathways in T cells. So in an attempt to address this question or answer this question, I'm going to ask the following questions and answer them as well. So what is the function of AIF in lymphocytes? Why is AIF essential for survival of T cells but dispensable in B cells? What is the role of c-MYC in driving effector versus memory differentiation of T cells? And finally, does targeting metabolism influence CAR T cell differentiation? As all of you know, there's two main pathways in the cells to produce energy, and there's glycolysis and oxidative phosphorylation. So I'm just going to give a very brief overview of those two processes. During glycolysis, glucose is converted to pyruvate, which can have one of two fates. It can be either converted to lactate and excreted, or to acetyl-CoA, which then fuels the TCA cycle. The TCA cycle has two main roles. It supplies substrates for synthesis like citrate, but it also produces the reducing equivalents necessary to support oxidative phosphorylation. So during oxidative phosphorylation, electrons are passed from an electron donor to a more electronegative acceptor, and oxygen is the final electron acceptor, which is then reduced to water. And when electrons are passed between a donor and an acceptor, free energy is released, and this free energy is then utilized by complex 1, 3, and 4 of the respiratory chain to pump protons across the inner membrane from the matrix to the intermembrane space, leading to the generation of the mitochondrial membrane potential. And it's ultimately the proton gradient that then drives ATP production when those protons flow back through complex paths. AIF is a mitochondrially localized protein, and it was initially described to be the main mediator of caspase independent death, as it was observed that upon permeabilization of the outer mitochondrial membrane, AIF translocates through the cytosol to the nucleus, where it then induces chromatin condensation as well as large-scale DNA fragmentation, actively leading to the death of the cell, and therefore it was also named AIF for apoptosis-inducing factor. However, very soon after its initial discovery, it was also observed that loss of AIF leads to loss of expression of respiratory chain complexes within the mitochondria, which then in turn leads to defects in respiration and an increase in ROS, causing the death of the cell due to metabolic insufficiency. So I was therefore interested to understand the role of AIF in lymphocytes. Is it actually an active mediator of cell death, or is it important to maintain survival of those cells? AIF is embryonically lethal in constitutive knockout animals. So I therefore crossed a conditional AIF locked animal first to a lock-stop locked YFP report animal. And this allows me to track the cells that have successfully deleted AIF by just following the YFP signal. And those animals I then crossed to tissue-specific deletion animals, where I then deleted the AIF specifically within the B cells by crossing it either to the CD19 Cree mouse, or I crossed it to the LCK Cree mouse if I wanted to delete AIF only within the T cell lineage. To understand the role of AIF within mature T cells, the first thing I did is that I isolated the spleens from wild-type or T cell specific AIF knockout animals. And what I observed is that loss of AIF leads to a severe reduction of those T cells within the periphery, and this effect is always more pronounced on CD8 T cells compared to CD4 T cells. And that is something I consistently saw throughout my studies. And as has been described in the literature before, loss of AIF leads to reduced expression of subunits of respiratory chain complex 1, 3, and 4, but not complex 2 and 5. And the most likely explanation for this is that complex 1, 3, and 4 form those so-called supercomplexes. And it's thought that loss of one part of the complex leads to disintegration of the whole complex and then to the degradation of all of its components. And even though loss of AIF reduces the amount of respiratory chain complexes within T cells, it does not affect the ability of T cells to consume oxygen under resting conditions. However, it dramatically impairs the ability of those T cells to upregulate oxophosphorylation in response to oxidation. I therefore wondered if one possible explanation for the fact that I see a lower frequency of T cells in response to deletion of AIF is that a loss of AIF causes an impaired homeostatic expansion. So to test this, I isolated T cells from either a wild type or T cell specific AIF knockout animal, and I then injected them at a 1-to-1 ratio with congenic T cells into a REC1 knockout mouse. And indeed, loss of AIF impairs the ability of T cells to homeostatically expand. And again, this effect is more severe for CD8 T cells than for CD4 T cells. Having established that AIF plays an essential role in maintaining homeostatic expansion and survival in T cells, I next wanted to investigate the role of AIF in mature B cells, and because I wondered if it's equally important for the maintenance of those cell populations. So I therefore, again, isolated spleens, but this time from wild type or B cell specific AIF knockout animals. But what I observed now is that loss of AIF does not affect the number of B cells within the periphery, and there's also no selection against the loss of AIF, as I don't see selection against loss of YFP. And just like what I observed in T cells, loss of AIF still leads to the reduction of subunits of complex 1, 3, and 4. But in contrast to T cells, loss of AIF does not impair the ability of B cells to consume oxygen, either under resting conditions or in response to activation. And I have to say, I was a bit surprised by those findings because I have to admit, I did not expect to not see any defects on B cell function or survival if I see such a reduction in expression of those respiratory chain complexes. So I thought, okay, if I just use a stringent enough model, then I will be able to detect a defect in those B cells that have deleted AIF. So for this, I infected mice, either wild type or B cell specific knockout mice with flu. And then on day 10, I analyzed the number of antigen-specific B cells, either within the mediastinal lymph node or the spleen. But even under those conditions, I could not detect any defects within B cells due to loss of AIF. So this leads to the obvious next question. Why is AIF essential for survival of T cells, but completely dispensable in B cells? There have been reports in the literature describing that for T cells to mature out of the thymus, they need to delete the mitochondrial content via autophagy or decrease the mitochondrial content. So therefore, the idea is that if they have reduced mitochondria to start off with, perturbation will have a more severe effect on them. So to test this, I isolated B and T cells from a wild-type Labroots animal and determined the amount of mitochondria within each of those populations. However, as is shown here, those B cells and T cells have comparable amounts of mitochondria, so most likely that's not the explanation. But since AIF is a mitochondrially localized protein, I wondered if maybe there's a qualitative difference between the mitochondria in T and B cells. So I therefore took advantage of the cationic dye telomerase. So the uptake of this dye is directly depending on the mitochondrial membrane potential. So the higher the mitochondrial membrane potential, the more dye will be taken up. And indeed, what I observed is that CD8 T cells have the lowest mitochondrial membrane potential, whereas wild-type B cells have the highest mitochondrial membrane potential. And this is obviously really interesting because this is an inverse correlation to the dependence on AIF. To understand the role that this mitochondrial membrane potential plays within those two cell types, I repeated this experiment, but this time in the presence of oligomycin. So oligomycin is an inhibitor of complex 5 and therefore prevents the flow of protons back from the intermembrane space to the matrix. So if the cell now uses the oxyphosphorylation to generate energy, that would mean that complex 1, 3, and 4 still pump protons across, and that would lead to an increased mitochondrial membrane potential. And this is indeed what I observed in case of the T cells. So now oligomycin increases the mitochondrial membrane potential to similar levels observed for B cells, whereas oligomycin has no effect on the mitochondrial membrane potential in B cells, giving the idea that most likely B cells rely on glycolysis to generate energy. Another way to test this idea is by modulating the available carbon source in the media. During glycolysis, glucose is converted to glucose 6-phosphate, which can then either be shuttled into the pentose phosphate pathway to produce reducing equivalents as well as nucleotides, or it can ultimately be converted to acetyl-CoA, driving the TCA cycle. In this scenario, the T cell or the B cell can produce ATP via glycolysis as well as oxidative phosphorylation. However, if you now switch from glucose to galactose, galactose is also converted to glucose 6-phosphate by the Labroots pathway, but this is a very slow process, and therefore most, if not all, of the glucose 6-phosphate is now shuttled into the pentose phosphate pathway, and therefore the cell will not be able to produce energy via glycolysis. However, oxidative phosphorylation can still go on as the TCA cycle can be replenished via glutaminolysis. I therefore tested the ability of wild type B or T cells to proliferate in either the presence of glucose or galactose, and whereas T cells are still able to proliferate in galactose-containing media, even though it's at a slow rate, B cells are unable to do so, again indicating that they most likely rely on glycolysis to generate energy. And it's also therefore not surprising to see that since loss of AIF impairs mitochondrial function, loss of AIF also impairs the ability of T cells to proliferate in galactose-containing media, as is shown here. And again, this effect is stronger for CD8 T cells than for CD4 T cells. So consequently, loss of AIF leads to reduced ATP generation within the T cell population, but not within the B cell population. So just to summarize this part of the talk, what I've shown you is that in healthy cells, when AIF is expressed in the mitochondria, B cells relying on glycolysis, as well as T cells relying on oxidative phosphorylation, can generate the energy they need and are therefore able to survive. However, depletion of AIF leads to reduced expression of respiratory chain complexes within the mitochondria. So now B cells, since they rely on glycolysis to generate energy, are fine, whereas T cells are now unable to meet the metabolic demands and will therefore die. In the next part of this webinar, I'm now going to talk about the role of c-MYC in driving effector versus memory differentiation in T cells. Our lab has previously shown that activation of T cells leads to an upregulation of c-MYC protein within the first 24 hours. And it's this upregulation of c-MYC that ultimately drives this metabolic reprogramming, as deletion of MYC leads to reduced glycolysis. And as I've said in the introduction, this metabolic switch to aerobic glycolysis is essential to support exponential growth and proliferation, and therefore loss of MYC also impairs the ability of T cells to proliferate. Having established that MYC plays an instrumental role in regulating metabolic programs at the early stages of activation, we next wanted to understand if MYC is equally important to maintain this metabolic program once the cells have started to divide. To visualize MYC in activated T cells, we isolated T cells from c-MYC GFP fusion animals that also express the OT1 transgene. We then in vitro activated those T cells with CD3, CD28, and gated on the CD8 high and CD8 low population within the first division, as CD8 is a marker for asymmetry, and observed that the cells that express high levels of CD8 also express higher levels of c-MYC. We then confirmed those results on a per cell basis by confocal microscopy, observing a really nice correlation between CD8 and c-MYC expression. And this indicates that c-MYC, just like CD8, is asymmetrically distributed in activated T cells. Those different levels of c-MYC also have a significant impact on the metabolic phenotype of the cells after division, as the c-MYC high cells have a higher rate of glycolysis compared to the c-MYC low cells, And this higher rate of glycolysis also drives enhanced proliferation in the c-Myc high cells. So this shows that the c-Myc level within the first division directly affects subsequent proliferation events in the T cells. To now understand the functional consequences of c-Myc expression in vivo, we adoptively transferred c-Myc high cells or low cells from the first division into congenic wild-type animals. And these animals we then infected with flu and analyzed the immune response 2 weeks later, or additionally, we re-challenged those mice after 4 weeks with a heterosubtypic virus to restrict the recall response to the T cell lineage only before analyzing the spleen and peripheral lymph nodes. During the primary response, we mainly detected the c-Myc high cells, which is not so surprising considering they have a higher proliferative capacity, whereas we barely detected the c-Myc low cells. However, in response to re-challenge, we observed the inverse pattern. Now, the c-Myc high cells were barely detectable, indicating that even though they had a robust effector function early on, they failed to differentiate into proper memory. In contrast to this, the c-Myc low expressing cells were able to persist and therefore could respond to secondary challenge. So if our hypothesis therefore is true that c-Myc, by regulating metabolic programs, directly regulates the function of the cell, then we should be able to affect the T cell phenotype by modulating c-Myc levels. So to test this idea, we adoptively transferred CD8 high or CD8 low cells isolated from either a wild type or a MYC het animals, which therefore has reduced expression of MYC. And in that scenario, the CD8 high cells isolated from a c-Myc het animal responded equally well to secondary challenge compared to the CD8 low cells from the het or the wild-type animal, whereas the CD8 high cells from the wild-type animals failed to do so. So based on this data and data I unfortunately don't have time to share with you today, we propose the following model. A strong activation stimulus for a T cell leads to increased surface expression of amino acid transporters, which then enables the uptake of amino acids, driving mTORC1 activation and c-Myc expression, which then in a positive feedback loop leads to further enhancement of surface expression of those amino acid transporters, driving the generation of T cells with an effector-like phenotype. On the other hand, if the T cell receives a weaker activation stimulus, a lower amount of amino acid transporters will be expressed at the plasma membrane. Therefore, less amino acids will be taken up, leading to reduced mTORC1 activation or c-Myc expression, and this drives the generation of T cells with a memory-like phenotype. Because the T cell memory effector and differentiation is regulated by metabolism-driven processes, the approach that by manipulating metabolism, you can generate an effective as well as sustained anti-tumor response is really appealing. Whereas increased glycolysis leads to the generation of effector T cells, it often also drives terminal differentiation. On the other hand, reduction of glycolysis or inhibition of glycolysis drives the generation of T cells with a more memory-like phenotype that are able to persist and therefore respond to tumor relapse. So at this point, to test this approach and even implement it, and to understand if by targeting metabolism we can influence the CAR T cell effector versus memory differentiation, I decided to move from academia to industry, and first to Bluebird and now to Elevatebio. So we at Elevatebio pursue a three-pronged approach, focusing on innovation, enabling technologies, and also building out the necessary infrastructure for the development of the next generation cell and gene therapies. And we at Elevatebio strongly believe that the C-host technology is going to be very useful for us and helpful to achieve our goal, as it can be applied across multiple platforms during the development of cell-based therapies. So not only in research to understand how manipulation of metabolism can drive a desired phenotype, but also in cellular process development to measure the effect of process or process change on T-cell metabolism, as well as in correlative sciences to understand the correlation between T-cell phenotype and response to immunotherapy, as this is essential for us to be able to develop successful therapies. So unfortunately, I'm not able to share any internal data with you today. So I'm going to highlight data from the literature to support the idea that targeting metabolism is a valid approach in the development of cell-based therapies. Mellenfors and colleagues published really convincing data showing that it's actually the intrinsic properties of the CAR T cells themselves that's the most predictive factor in determining clinical response. So what they did is they analyzed 41 samples from patients with high-risk CLL that had received at least one infusion of CD19 targeted CAR T cells. And they observed that CAR T cells isolated from complete responders had high expression of genes driving memory, particularly early memory differentiation, whereas CAR T cells isolated from non-responders had increased expression of genes regulating glycolysis as well as exhaustion. So based on this data, since a more memory-like phenotype seems to be responding or corresponding to a positive clinical response, how can we use this information and modify or engineer our CAR T cells in such a way to drive memory differentiation? So one approach would be to directly focus on the co-stimulation provided during the expansion process, as it was shown that adoptive transfer of OT1 T cells into mice followed by priming with either over anti-CD27 or over anti-4-1BB had very different effects on the CD8 T cell memory versus effector differentiation. CD8 T cells primed with 4-1BB produced lower amounts of the effector cytokines perforin and IL-2 in response to re-challenge ex vivo. However, despite having a lower effector response early on, those T cells were able to differentiate into functional memory, as a higher frequency of them could be detected in multiple tissues, including lung, liver, and spleen, 57 days after the initial transfer. One possible mechanism how 4-1BB agonism drives memory differentiation is by increasing mitochondrial function. So as is shown here, activation of T cells with anti-CD3 and anti-4-1BB for 24 hours, followed by 7-day expansion in IL-2, leads to an increased mitochondrial content within those T cells, as shown here by FACS. And whereas 4-1BB agonism or stimulation does not affect the basal oxygen consumption, it profoundly increases the respiratory capacity of those cells, driving a metabolic phenotype that's associated with memory T cells. In 2016, Carl June and his team published really nice work showing that the choice of co-stimulation domain within the CAR itself can have a profound effect on the metabolic phenotype of the CAR T cell and therefore on the CAR T cell function. So what they showed is that inclusion of 4-1BB co-stimulation within the CAR design leads to an increased mitochondrial biogenesis, driving increased oxygen consumption under basal conditions, as well as increasing the mitochondrial capacity overall, and therefore leading to the generation of T cells with a central memory-like phenotype. And it's this phenotype that's positively correlated with clinical response. And I was actually really excited to see that discoveries made in basic research can be translated into a therapeutic setting. So just to summarize what I've shown you today, I've shown you work describing that immune cells, including B and T cells, have different metabolic requirements for survival and function. Also, that effector and memory T cell differentiation is regulated by metabolism-driven processes, that clinical responses in CLL anyway are determined by intrinsic programs of memory versus exhaustion. And finally, that the choice of co-stimulation can have a profound effect on CAR T cell function. So I therefore strongly believe that manipulating T cell metabolism is a really promising approach to generate T cells with immediate anti-tumor function, but that can then also convert to memory T cells for a sustained immune-mediated anti-tumor response. Our knowledge has grown exponentially within the last couple of years. However, I think there's still quite a few things we need to understand for us to be successful in implementing this approach and the design of cell therapies. So these are just a few, obviously, of the open questions I'm curious to address. So we still need to understand what is more efficient in generating an effective and persistent anti-tumor response. Is it boosting oxidative phosphorylation? Is it dampening glycolysis? Or is it actually boosting both of them? But if we were to boost both of them, what would it do to a pretty much used up T cell at this point? Based on the central role c-Myc has in driving metabolic reprogramming, I think it's also a really interesting target to consider. However, would the negative impact on proliferation outweigh the benefit of increased oxidative metabolism and persistence? And what is the effect of c-Myc deletion on T cell functionality? Would it affect cytokine release? And I think another big area where we really need to gain more understanding is what is the influence on the cancer biology on the T cell biology? So is it possible to override any type of tumor biology by just armoring the CAR T cells? Or do we have to consider targeting the cancer at the same time to get the optimal response? And then also, do we have, and most likely, match the modification to the tumor type? And what modifications would be more efficient? So I finally, last but not least, obviously would like to acknowledge the people that supported me throughout my scientific endeavors. And that's first and foremost Doug Green, who was a great mentor and actually whose curiosity for science is pretty infectious. And then my colleagues at Bluebird Bio, where I made the first foray into cell therapy. And obviously, my current colleagues at Elevatebio, who share the same passion as I do for targeting metabolism in designing the next generation of cell-based therapies. And lastly, I would like to thank all of you for taking the time to listen to my seminar today. So thank you.
Thank you so much, Sandra. That was an amazing presentation. We had lots of people watching, and there's lots of questions. So we will now start the question and answer portion of the webinar. Again, if you have any question that you'd like to ask, please submit it now. We'll be able to get a couple of questions in before the end of the hour. However, even if we don't make it to your question, we'll be forwarding all of them onto Sandra and the Agilent team to follow up with you. Additionally, if any of you would like to learn more about the solutions that we offer for cell analysis, you can follow the links on the bottom left section of your screen under the resource list to our new immuno-oncology and immunotherapy web pages. Let's get started. So the first question, Sandra, is there currently any approved therapy or even maybe therapy in development that you're aware of that includes CAR plus a metabolic gene alteration to promote cell persistence?
So I know that there's some efforts implementing those approaches, but I have to say, I mean, I switched to industry about 3 years ago, and considering the strong support in academia, I think industry is a bit slow to pick it up. I would have expected to see more by now, but yes, there are some efforts, but they're all preclinical, and I don't know of anything being already in the clinic.
Okay, thank you. We have a question here relating to c-Myc. The question is, how does c-Myc impact the potency of T cells? Is there a balance that should be achieved between T cell fitness and T cell potency?
Oh, I think this is a great question. And we always know that obviously driving a persistent response is essential to have a successful immunotherapy-based target. However, you don't want to swing the pendulum too much because you really need this potent anti-effector response, anti-tumor effector response early on. So to strike the right balance is actually going to be really hard, I think, and it's something that we have to be very cautious about, that we don't overdo it with the memory, because if we don't get immediate tumor clearance early on, then it doesn't help us if we have memory cells. So that's why we really need to think about how can we stimulate the T cells or modify them or engineer them in a way to maintain this plasticity that gives us this really good punch early on, while then also giving us the persistence. And I obviously don't have the answer for this, and the data has to bear it out, but maybe we have to have a two-pronged approach. And if we want to push them to really strong effectors at one end, maybe there will never be really good memory. So we have to then think about, do we go in with two different populations to start off with? But it's a great question.
Great, thank you very much, Sandra. mTOR is also asymmetrically distributed in effector and memory T cells. Do you think that that could be a link to asymmetric activation of c-Myc in these cells?
Oh, absolutely. So we obviously did work on mTOR, so did Jonathan Powell. So I just cut it out due to the time constraints. Yes, I think mTOR is a very attractive target and one I would definitely pursue. So yes, absolutely. And it might actually be easier to regulate mTOR1 levels than trying to modulate and hit the sweet spot with targeting MYC.
Sandra, maybe you could, just thinking about solid tumors and the tumor microenvironment, we have a question here which is, I suppose, asking your opinion about whether shaping CAR T-cell metabolism, how challenging that will be in solid tumors because of their heterogeneity.
I mean, I think that's a great question. And my general thoughts are, yes, I think definitely you can arm the T cells in such a way to obviously make them at least competitive, because I think that's a problem with the tumor. They need the same nutrients. They drive very similar types of metabolism, but somehow a tumor cell is always better, which is a shame. So I think, so for this, again, if you give the tumor cells time to adapt, I think you will lose that rate every single time. So I think, yes, you have to arm the CAR T cell to be successful early on. And yes, I think you have to enable it to successfully compete against it. And I mean, there's really strong work by Erica Pierce showing that, depending on the glycolytic dependence of the tumor, that really affects your T cell response. So yes, so you have to target it. And again, for this is what I try to get at. You also need to understand the tumor biology so that you can kind of, tailor your T cell to the tumor you like to target. And maybe, as I said, you can hit the tumor at the same time. So that's the nice thing about cellular therapy, right? You take out the cells, so you can engineer them. And therefore, you can maybe add drugs that, inhibit glycolysis, stop tumor growth. And then you can engineer T cells to be resistant to those drugs, or, to arm them that they now can compete better. So I think there's a lot of things you can do in this area, which is why I think it's really exciting. So the possibilities are endless, but to identify which is one the most successful one, I think that's a challenge.
Yeah, I couldn't agree with you more. So regarding just comparing tumor cells and immune cells, which in your experience are more sensitive to amino acid deficiency?
Oh, that's a good question. And I actually don't know. I would think, and again, sorry, I'm just speculating, tumor cells tend to be very adaptable to changing conditions, which is why they survive so well. So I think if put under the same strain, that tumor cells would be able to utilize other pathways better than T cells do. Because, for example, lactate, they can cope with high lactate, they can take it out and use it for their advantage, whereas T cells really don't do so well in that environment. So I actually think, having said that, if you put strain on and, metabolic stress on cells, tumor cells will find a way around it much better than T cells would.
Yeah, that's a good question, isn't it? Maybe somebody in the audience will put that into the chat. So the next question then, so which of these is practical? So screening or isolation of less glycolytic CAR T from a production, or pharmacological or genetic modulation of metabolism in CAR T cell design or propagation?
So I think it's a two-stage process. So yes, you want to have the cells. I mean, everyone talks about now more Mary-like or less differentiated, so that they have this plasticity early on. So I think, so therefore, to, either adapt your process so that you tend to increase the percentage of those cells during expansion, or that you isolate those less differentiated cells early on. I think that's one part of the puzzle. I think the other part you have to solve is, once the T cell encounters the tumor, I think you still need to modify the T cell in such a way to basically be immune. I mean, it's an immune cell, but to resist this very stringent tumor microenvironment. So I think it's two things you need to solve separately.
Thank you. Coming back to metabolism, and you're obviously a huge fan of metabolism in the study of metabolic parameters for immunotherapy. But do you think that it's going to be a key component for immunotherapy success in solid tumors? And do you think the impact of that will ultimately be higher than for blood tumors?
So yes, so the answer is obviously yes and yes and yes, but that's just, I'm as you said, I'm a bit biased. So I'm not sure it is for. So let's go to solid tumors. I definitely think that targeting metabolism is going to be important at driving an effective immune-mediated response. And I always like to take the, as I call it, the kitchen sink approach. Throw in as much as you can to get the best chances early on, because you don't want the tumor to give the tumor time to adjust. So you want to give your big punch right away. And I think, so therefore yes, metabolism is going to be an essential component of this. However, I still think, yes, it's not quite the same stringency you have in a heme tumor. So you still have, obviously, more nutrients available. But I still think that there's going to be some what you learn out of a solid tumor that can be translated to heme tumors, and then even make the T-cell responses better against them. Because even in heme tumors, right, it's interesting, not every heme tumor responds equally well to CD19 CAR T-cells. So there's definitely a lot of, especially CLL, I think the response rate is 20%. So there's still a lot you can do to increase the number of patients benefiting from this therapy.
And Sandra, just continuing on the theme of kind of a broad view of immunotherapy, can you take a few minutes just to discuss, the state of the field and the direction that you feel for immunotherapy, where it's going, and at what point you think it'll be truly adopted for clinical treatment, and what are some of the, in your view, some of the major gaps or major challenges ahead of us?
Oh, that's a very broad question. So that's really interesting. I can try to tackle it. So I think you see the potential, right? If it works phenomenally. But if it doesn't work, we still don't always understand why it doesn't work. So we really need to get a better understanding of why it doesn't work. And then there's also, treatments out there or indications. So solid tumor, we still haven't tackled. And then the biggest problem is also, it's hard enough to find one. So one is the limitation, obviously, of a response to it, but then also to identify those antigens. It's also obviously another challenge. And then you think about the tumor heterogeneity that needs to be addressed for a solid tumor. So it's a big question. So I think the next area is going to be solid tumors to see this, but it needs to be in context with targeting the tumor heterogeneity. So I don't think, and again, that's just my opinion here, I don't think CAR cell therapy can be a single modality in certain settings. So I think to think about how to incorporate it into current treatments, that's great, but the potential, I think, is really enormous. I would say CAR, but that would obviously include also TCR therapy. And I think the next level is just like and that's what we think about, right, at Elevate. So yes, the costs are really high right now, which are prohibiting to make it widely available. But how about if you improve manufacturing? Can we drive down the costs? If you move away from, like using viruses to produce the cells, can you do in vivo editing? And can you also, or think about an allo approach so that you drive down the cost of the product? And I think this a lot of times will then actually make it more wildly applicable. And I think you also have to see, so far, the other thing is, I think what would be a big sticking point for those type of therapies if it actually was ending up to be a cure. That would obviously be such a game changer compared to, where a lot of treatments right now are treatments and they, will benefit the patients, but the relapse rate is really high and they are not considered cured. So this is where I think that's my 2 cents for that area.
Thank you, Sandra. It was a very broad question, and you did a great job of answering that.
Thank you.
We're coming up to the close of the hour, so just a couple of, and it's great that we've had time for so many questions, and we have lots more actually that we'll send on to you. But a couple of smaller questions just before we come up to the hour. We talked about memory T cells and that being, one of the goals of your work. What do you consider are some of the best markers for looking at memory T cells?
Yeah, I mean, it's not just memory, right? You want the early memory, so you want TSCM or TCM over TEM, right? So just to even stratify there a bit. So I like, and again, I mean, it's just a few. There's really nice panels out there. I think it depends on how far you want to go, but CD45RO, CD45RA, and CCR7, and if you want to include CD62L and CD95, actually give you a really nice coverage of most of those phenotypes.
Thank you. Just one last one then. So earlier in your talk, you talked about AIF and the difference between T cells and B cells, but did you find that the AIF knockout, did you look at or could you project in terms of what effect that would have on memory?
No, I mean, unfortunately, I didn't, because the AIF knockouts, they don't persist. So if you were to challenge them or in an infection, I don't think they cannot expand. So they cannot go through the effector phase. So they wouldn't form proper memory either.
Okay, thank you. Sandra, thank you again for an amazing presentation. And as you did as well, I'd like to thank our excellent audience and the fantastic questions that we've had today. So for everybody on the call, until next time, thank you and goodbye, everyone.
Thank you for leading through this discussion. That was really, really, really exciting. Thank you so much.
Thank you.